Good day to everyone joining us, and welcome to today's X-Talks webinar. Today's talk is entitled Technology Selection for Enhancing Bioavailability. My name is Andrew Jernan, and I'll be your X-Talks host for today. Today's webinar will run for approximately 60 minutes, and this presentation includes a Q&A session with our speaker. This webinar is designed to be interactive, so please feel free to submit questions and comments for our speaker throughout the presentation using the questions chat box, and we'll try to attend to your questions during the Q&A session. This chat box is located in the control panel on the right-hand side of your screen, and if you require any assistance, please contact me at any time by sending a message using this chat panel. And at this time, all participants are in a listen-only mode, and the presentation slides will advance automatically for you. Please note that this event will be recorded and made available for future download. And at this point, I would like to thank Bend Research, who has helped develop the content for this presentation. For more than 35 years, Bend Research has worked with clients to create value by advancing new medicines that improve human health and to solve their most difficult scientific and technical problems. This success is based on the company's ability to develop, advance, and commercialize pharmaceutical technologies, which grow from a solid base of scientific and engineering fundamental understanding. Bend Research is a leader in novel formulations, including spray-dried dispersion and hot melt extrusion formulations, as well as controlled release, inhalation, and biotherapeutics technologies. Bend Research is now part of Capsugel's dosage form solutions business unit headquartered in Morristown, New Jersey, providing access to additional technologies, R&D capabilities, and global manufacturing infrastructure for the advancement of client compounds from concept through clinical and commercial manufacture. Bend Research has more than 250 employees based in 6 state-of-the-art facilities in Bend, Oregon, and is part of the global Capsugel network of 3,100 employees. And now I would like to introduce our speaker for today's event, and that is David Vodak, Ph.D. Dr. Vodak is Vice President of Chemistry at Bend Research and has been with the company since 2004. His past positions have included Director of Research and Director of Physical and Biological Sciences. Dr. Vodak's areas of expertise at Bend Research include the research and development of novel pharmaceutical drug delivery systems with a focus on the delivery of low solubility compounds and on a pulmonary platform based on spray-dried engineering particles. His other research has centered on the characterization and investigation of physical properties, stability, and solubility of technologies aimed at enhancing bioavailability, such as spray-dried dispersions, high-energy nanocrystals, nanoadsorbents, nanoparticles, and spray-layered dispersions. Dr. Vodak holds a Ph.D. in materials chemistry from the University of Michigan and a BA in chemistry from Willamette University. He is a member of the American Chemical Society and the American Association of Pharmaceutical Scientists, and he holds 3 patent applications. And now, without further ado, I would like to hand the mic over to Dr. Vodak, and you can begin your presentation when ready.
All right, well, thank you very much for the introduction, and I'd just like to say a quick thank you to everyone that's online for tuning in. The presentation, this presentation will be the first in a series of webinars that Dosage Form Solutions will be airing over the next year. And the purpose of this presentation is to sort of be a survey of what are the types of technologies that are available and how to think about mating those technologies up with the right compounds based on physical properties and some other things. So today we'll introduce the topic of the technology selection for enhancing bioavailability. And as you'll see, this is certainly a work in progress, but I think it's important to get this out here. This is one of the more important things in the area in terms of being more efficient on the front end. So for the follow-on seminars, we'll then take deep scientific dives into each of the technologies that we discuss, and that'll support this higher-level survey and overview that will be covered today, which is a good lead into the next slide. as to what I hope to cover this morning. So I think the introduction already covered quite a bit of what I was wanting to make sure that everyone had background on in terms of the capsugel, dosage form solutions, and Ben research relationship, but may touch on a few points there, and then get right into the technical content and really kind of talk about the problem statement that we're dealing with, do a real high-level survey of kind of all of the technologies or many of the technologies that are available for solving these problems, and then take a step down and get more into some of the specific technologies that we have specific experience dealing with, and talk about what kinds of compounds and product concepts are typically appropriate for each of those, and then get into some guidance maps, and certainly these are fairly qualitative and conceptual at this point, but our hope is as we continue to interact with our collaborators and our colleagues in DFS that we can build these into more quantitative. And so today it's really more just to kind of express how we're thinking about the idea of technology selection. And then as usual, just to kind of highlight that process, we'll go into a few case studies where multiple technologies have been selected and how those have been used to enhance and enable programs. So to start with, there's just a brief introduction. It's very exciting news for us earlier this year that Ben was acquired by Capsugel and merged into the Dosage Form Solutions division to be a part of a broader technology experience base. And basically, what does this mean for Ben Research? Well, as you heard, not much has changed. We still are pride ourselves in having a reputation in scientific and engineering excellence. Have about 250 folks here in Central Oregon, and as will be a theme of this presentation, the focus is on solving hard problems here as part of helping to enable products to progress through the drug development process. In terms of dosage form solutions and with Ben Research, you can see these different areas and all the technologies and capabilities that are present in this larger integrated entity. And as a part of this offering, there's a wide range of technologies, but the idea here is still not to vend the technologies, but rather just to have this integrated place where compounds can come and you can interact with the experts in each of these areas to essentially decide based on your product concept what the right technology solution is, and also have the downstream commercial and clinical supply manufacturing infrastructure to back up these technologies so that you have a path forward. Okay, and with that brief introduction, I'd like to get into just kind of talking about what the problem statements that we're here to solve today or talk about solving. So given all the technologies available, today I'm going to focus on the bioenhancement and problem statements where we're talking about poorly soluble compounds. And this is a very familiar plot to people in the area. And I think not to spend too much time here, but one of the main points to make is that the portfolios that we're seeing and interacting with are driving towards DCS2B, definitely the DCS2 area, but more specifically driving towards DCS2B and DCS4. So that means that more and more often compounds are dissolution rate and solubility limited for absorption, and that the technologies required to enable these compounds in these spaces will need to achieve not only improved dissolution rates, but also supersaturated dissolved drug levels relative to lower energy crystalline forms. Of course, there are many enabling technologies available, almost too many. So the idea is to select the best approach and get to that as soon as possible so that you're not spending extra resource and time going down dead ends. And I think that's really what the mission of this presentation is about. And it's definitely a work in progress, but I think it's a worthwhile goal to have in mind. And so when you're selecting the technology, what's the best for your specific molecule? And that means, your program. So not just API properties, but things like dose and product concept. And I'll talk a little bit more about some anecdotes we have of, maybe the compound was perfect for a certain technology, but because of something marketing wanted or something that we needed for the therapeutic in terms of a combination, another technology suddenly became more appropriate. So the way I've divided up to kind of talk through these different technologies here are kind of thinking about things in terms of solid state properties and particle size, and these technologies tend to be more in the metastable regime in terms of a higher energy form. And then on the other side, we're talking about things where things are dissolved. So solvation or dissolved drug is the actual dosage form, and these tend to be more in the thermodynamic regime. And then in the overlay, we have things that are kind of somewhere in between metastable and thermodynamic. For example, melt extrudates oftentimes work more towards a thermodynamically miscible system, so it would be more, even though it's a solid state, it would be more like a dissolved system. And for anyone that wants to really dive in and get a really, really nice, complete overview of a lot of different technologies for bioenhancement, I recommend this recently published review by Huell Williams et al. It's very complete. All right, so just to start off with, what are ways that we can formalize how to select technologies based on API properties, just as an initial starting point? We like to use guidance maps where you plot different API properties against each other and then overlay that. So here the gray dots are actual API programs that we've developed, and you kind of look and say what technologies were appropriate for these. This is certainly a very simplistic and conceptual map, as this is a multi-dimensional problem, but just for starters, let's wade in. And so this plot is showing aqueous solubility as a function of LogP for a dose of about 100 milligrams, I think. And as I said, the gray points are actual compounds, and the shaded regions show where, by experience, we had success with these molecules. So I'm going to come back to this after we cover a bit more detail on the varying technologies, but I wanted to introduce it here so that when we see it later, we have some context. So first technology I like to talk about is a very well-known technology for improving solubility of low aqueous solubility compounds, and that's using a higher energy salt form. And this is very well precedented. A large percentage of marketed products utilize this approach. And so the compounds that are typically applicable for this type of technology obviously are ones that do need some solubility enhancement. So DCS 2B, 2A and B, or 4 may also give you some help. Weakly ionic, basic, or acidic. We see a lot of weakly ionic, weakly basic compounds that require, and typically in this case, crystalline is a benefit, just in terms of being able to have that potential advantage of that's your drug product. And so some of the advantages associated with this technology are obviously a high dose potential. We've had a lot of success combining this approach with other technologies, as I'll talk about later in one of the case studies. Lots of precedents, fewer unit operations, and amenable for solid dosage forms, even capsules or tablets. Some of the challenges are common ion effect. We're making a supersaturated state, and so we can have tendencies for precipitation going from, for let's say, a weakly basic compound going from stomach pH to intestinal pH, and then we've also observed conversion to lower energy form and a higher gastric pH, which from a product concept would oftentimes mean you're going to have some sort of a label like don't take with proton pump inhibitors. And then, obviously if you get the salt enough, or if the salt is too large, now you're dealing with a salt factor which can compromise loading. But this is a very good technology, and as I said, I think has a lot of promise in combinations with other enabling technologies that we'll talk about. Another technology that we deal with quite often is the idea of nanocrystals, and mostly we deal with the top down, where we're using attrition or milling to generate sub 500 nanometer crystals to give the dissolution rate enhancement. We've also been dabbling in bottom-up nucleation of crystals, and that's a more well-controlled system in some cases, and actually has the potential to lead to very small crystals where you're actually in a higher energy regime and you can see solubility enhancement. But in general, for attrition, which is by far the more precedented route, we're typically looking at compounds that require dissolution rate enhancement only. It's the 2 to 500 nanometer regime is most specifically about surface area enhancement. And typically neutral or weakly ionic unionized compounds that have low aqueous solubilities. The attrition is typically done in a wet mill and aqueous environment, and if you have too much solubility in that environment, you just get ripening. It's very hard to make and establish that high surface area. And then typically, as I'll get into later in terms of maps, things that are crystalline, where we're looking at things that have medium to high melting points. I've got a schematic here talking through the different ways that you can make the nanocrystal. There's the milling. You can homogenize, or you can use like a jet precipitation, and in that case, that might be more of like a bottom-up nucleation approach. And then typically you can either take that suspension, and if it's stable enough and fits your product concept, the suspension can be what the product is, or if you'd like to convert to a dry powder, we can take those suspensions and do things like spray drying or lyophilization to get the dry powder and then get into your solid dosage form manufacturing. So the potential advantages here for this technology are, DCSA, so dissolution rate enhancement. There's a high dose potential. There's precedence in terms of multiple products on the market, and solid or liquid dosage form. Some of the challenges I mentioned: stability. You can get ripening if your solubility is too high, which leads to increasing in crystal size and kind of defeats the technology's purpose. There's typically little to no solubility enhancement. You may still have to deal with form changes, and then the cost of production is largely perceived to be relatively high, and there's a perception that there's a limited number of commercial partners. From the spray dry dispersion technology, one that we're very familiar with, this is a case where we're basically taking and making a kinetically stabilized high energy form of the drug that's stabilized by a high TG polymer, and using that to make a supersaturated state. And because of the particle size that you can enable and the metastable state that you've made, these are appropriate for DCS 2A, B, and 4. And I have this map here, and I'll go into more detail in a few slides, but in general, we have a pretty well understood formalism for selecting loading and polymer types based on a given API and product concept. And so we'll go into more detail on that. But so the idea here is we take an active, we dissolve it in a volatile organic with a high TG polymer. We atomize that out of the spray dryer and rapidly flash the solvent off to capture dry powder. They typically look like shriveled raisins, mostly due to the fact that the polymers that we're using can't move as fast as the organic solvent as it's retreating in the drying process. So you get a skinning, which that's the surface area, and then the polymers plastically deform as the rest of the solvent leaves, kind of shriveling up, making these raisin morphologies, and that's a tunable parameter. The product is typically characterized by a single glass transition temperature in a modulated DSC experiment, and then you look for things like supersaturation, sustainment of that, and then mate that up with either a suspension, a tablet, a capsule, even some modified controlled release forms are appropriate. So the advantages here that you get the solubility and dissolution rate enhancement. Spray dry process is well known to be a very scalable process. from milligrams to metric tons, and there's some precedence. There are a few products on the market now, and we see that kind of on the uptick. It's appropriate for solid dosage forms, applicable to large compound property space, and because we're breaking the crystal structure, you don't really have to worry about form issues. Some of the challenges are drug loading. We have to, oftentimes, depending on the properties, have to put quite a bit of polymer in to stabilize this. It's a solvent-based process, which you can get around, but sometimes that's problematic for certain facilities. The organic solvent solubility of your API may be limiting, so these compounds that are so-called brick dust may be hard to get enough dissolved in your organic to have a viable throughput in terms of a commercializable process. And then there's definitely a perception around physical stability, but I see the industry has kind of started to move through this and understand it very well, and I think the same thing can be said around, historically there was the perception there was a limited commercial partners, but even that started to kind of change, and even through DFS we'll be able to provide some level of commercial support in the coming years. So on the hot melt extrusion approach, here we look at a map, and again this is similar to the spray dried dispersion technology in that it's amorphous stabilized system with a polymer, although typically in this case, because of the process and the rate of quenching, we'll try to be more towards a thermodynamically miscible portion of the phase diagram, and therefore sometimes lower loading. And the compound properties that are important for HME are things like the melting point and how that interacts with heating in the presence of your polymer. So we have to get this melted and dispersed into it using this mixing process, and then further with the TG of the compound to decide on aspects of physical stability. And as I said, for the formulation considerations, we'll think about things like a binodal phase diagram and how we need to get up into a one phase regime during the process, and then as it quenches back down, where do we end up on the phase diagram? And here we just show some of the product, extrudate versus milled and chopped, and that's manipulated for your solid dosage form concept. Some of the advantages here, again, solubility and dissolution rate enhancement. It's a continuous process, very high throughput. Precedents, there are products on the market. It's for solid dosage form, and it's an extremely small process footprint. Some of the challenges are even more so than SDDs, drug loading, again, because we're trying to target low solubility actives having solubility in the polymer. Predicting physical stability can be challenged, because we're more towards a thermodynamic miscibility, setting things up at accelerated conditions to test how fast they fail. You're changing the phase diagram, and so it's harder to say what at an ICH type stability condition what you could learn about storage at that condition at an accelerated one. There's an upper limit towards the melting point in terms of compounds that aren't applicable, and obviously any compound that's got some thermal sensitivity becomes challenging with this. And then there's a perception that the scale down a process, so getting down into sub 1 gram in order to do feasibility. But I think things that have been shown out in the literature in the past few years have shown that that certainly is something that people are able to get around these days. So now a few words about lipid-based formulation. So what are the types of compounds that are appropriate? Well, we talk about compounds that have high LogP, kind of lipophilic, hydrophobic properties. And just looking at the polar surface area, dose is something that you want to think about here. And in general, compounds that this technology can be specifically enabling for compounds that have metabolism issues or efflux or permeability, and a lot of that is due to how these drug delivery systems work in terms of they not only provide, in cases where the formulation is tuned, not only provide just a physical supersaturation enhancement, but they also have methods of interacting with the actual physiology in terms of how the digestion and the biosalt micelles and the absorption of the lipids that you're putting in, and how that interacts with, say, the gut-associated lymphatic tissue and drug absorption through that route. So one of the really nice things about the lipid-based formulations is there's a wide range of excipients to select from. And so that means that you have the opportunity to really tailor your vehicle for the API. That's a really big positive. The flip side of that can be a little bit of a negative, is that maybe there are too many to choose from, and sometimes that can lead to an empirical approach. Capsugel, as they presented in a webinar recently, is in response to that, has developed an expert system that aids in coming up with a less empirical selection of the right excipients. And that's something that will probably be showcased more in a follow-on webinar. And then here just showing how two formulations, depending on how you formulate, we can end up with different particle sizes, and just the tunability that we have with this technology. I'm not going to spend a whole lot on this technology, but just in the spirit of what are some of the newer things that we're looking at to solve some of the problems we're seeing. And one of the technologies that's currently being investigated here, this is a concept that's been known and is scientifically valid, and here's one embodiment that we're currently evaluating for the following applications: bioavailability, chemical and physical drug stability, delayed sustained release, taste masking, odor masking, and especially around some of these peptides and oligonucleotides. So this is something that we're looking at right now, and we have some promising in vivo data that will show up in a case study showing bioavailability enhancement application. Okay, so that's the quick run through of the technologies, and so I'd like to just spend a few minutes and go through several guidance maps that we look at. And again, the point here is less to be very quantitative about this is exactly what you should go do, and more to show our process and our thoughts on the matter and how we're attempting to continually improve this. So this plot hopefully looks familiar from earlier in the presentation. And again, this is not a finished product. We're continually improving our ability to bin compounds, and this DFS opportunity, bringing experts on multiple technologies together is I think, going to really kickstart and help make this happen. So based on our experience, crystalline salts, amorphous, and lipid-based technologies are the most widely applicable, and there's a large overlap in the technology approaches. So typically it's a dimension not shown on this plot that further drives technology selection, so dose, product, concept. So, for example, one of the compounds that we had worked on, a compound called torcetrapib, made a very good soft gel lipid formulation, SEDS formulation, and that was great, and that was an ideal compound for that technology. However, the product concept was a fixed dose combo with Lipitor, and so we needed a dry powder form of torcetrapib in order to put into a single tablet. And so that's an example of how a compound can be perfect for a given technology, but there are some other factors, which is why this is so complicated, and why, or not complicated, but multidimensional, and why maybe this is still a work in progress. So so there's multiple technology formulation possibilities in each large group here, and then in order to not have a 12-dimensional plot, we've tried to subdivide each of the kind of areas and show maps of how in each area you might pick a formulation or a loading. So on slide 16, here's a conceptual nanoparticle drug formulation map. So compounds that are lower LogP but have very high melting points typically want to be crystalline, so we come up with technology approaches where they can be crystalline but in the highest energy state possible. And so here you're looking at things where you're going to get dissolution rate enhancement. For compounds that are more moderate to lower melting points, will do what we call drug-polymer dispersion nanoparticles, and there's a case study that I'll get to that describes that in a bit more detail. But those can be enabling for things like rapid onset, where dissolution rate's really important. And then, as well known in the literature, using either just pure amorphous drug for compounds that really have no driving force to be crystalline, or in the presence of some lipids and making nanoparticles out of those constructs. The spray-dried dispersion formulation guidance map, and this is probably more for amorphous dispersion selection. On the y-axis, we have driving force for crystallization, or the melting point to glass transition temperature ratio. And on the x-axis, we have the LogP, or the lipophilicity of the compound. And based on our historical experience, we can plot, essentially our scar tissue in the area of developing amorphous formulations and say, kind of draw in shaded areas of what worked and what didn't work, so that when a compound shows up, we can quickly and easily say, last time we saw a compound with these types of properties, what formulations were appropriate. And so compounds that tend to have a high TM over TG ratio, oftentimes we can have to suffer a lower loading because we need more polymer around to stabilize that state. And typically you're looking at formulations that require kind of 80 to 90% polymer here. It's also important to note that compounds that live up here tend to have very high supersaturation ratios in water, and almost always will require some sort of a sustainment precipitation inhibitor in the formulation. On the other side of things, when compounds get extraordinarily lipophilic, we tend to get limited in loading because you put so much of a greasy compound into a solid, the solid itself starts to behave like that greasy compound, and therefore you end up not being able to dissolve your particles. And so cases like this where you might, depending on the product concept, you might look for other technologies, things like high surface area or some of the lipid-based technologies. And then there's a very large space where you can generally look to formulate in the, anywhere from 90% drug loading to 50 to 25, which makes this a pretty good technology for a wide compound property space. Let's just say a few things on some of our thoughts around conceptual guidance for lipid formulations, and this is based on solubility in oils. It's a very simple general solubility equation kind of model, and basically what we're saying here is that the compounds that are ideally suited to this are relatively low dose, low melting point, and high LogP. And if we look at the conceptual map based on the general solubility equation, so this is just calculated based on solubility from that equation and LogP in a 0.6 mil volume. And this just conceptually shows how solubility tracks to dose based on a given melting point and a LogP relative. And I think it's also important to highlight here that despite the fact that we like to look at these things that are easily measured in terms of physical parameters, some of the things that these lipid-based technologies offer are a bit harder to measure as they interact with the physiology. So things like micelle-mediated transport through the mucous boundary layer, fed state induction, efflux inhibition or saturation, metabolic saturation, or lymphatically transported drug. So that kind of wraps up the guidance portion, and then I'd just like to dive into a few case studies here for the remaining 10 or 15 minutes, and then we can get to questions. So the first case study is high-energy crystals with a precipitation inhibiting polymer, and in this case, this is an example that came out of a U.S. patent that's been published, and the molecule is ziprasidone mesylate. It's a high-energy form of the drug ziprasidone, and what you see for just the crystalline ziprasidone mesylate is that it immediately converts to its lowest energy form, free base at this pH, and effectively you have zero solubility with this black line that you almost can't tell. And so in order to mitigate this, they looked at cyclodextrin complexes, and even that, you get a little bit of solubility enhancement with that, but not much here for the free base. And so one of the approaches that was tried here was to dissolve HPMCAS into the dissolution media, and then add in the ziprasidone mesylate cyclodextrin complexes. And what you see is that the HPMCS is catching the supersaturated state, and this is a very challenging compound, but you're still getting a great deal of supersaturation for the first 30 minutes in intestinal pH, and then even some enhancement here, probably relative for amorphous to crystalline. So it's a nice example of how you can take a compound, a high-energy salt that is going to supersaturate, and then combine it with a different or multiple different technologies to come up with a unique solution to your unique problem. The second case study here is based on a publication by my colleague Mike Morgan recently in PharmRes, and this was an example of amorphous drug polymer nanoparticles of a drug called celecoxib. And the product concept here was immediate release, as this is for pain, and so can we get even faster release than the marketed capsule? As you're thinking about amorphous systems, we pull out the TM over TG versus LogP plot, and celecoxib kind of lives right in here with these properties. And so we felt like the amorphous could be stabilized. How these nanoparticles were prepared, essentially celecoxib and ethylcellulose are dissolved into an aqueous immiscible solvent like dichloromethane or ethyl acetate, and mixed with a surfactant containing aqueous media, pre-emulsified with like a rotostator, and then run through a homogenizer to generate a nano emulsion, which then the organic solvent's removed and the product spray dried to get a dry powder. And if we look at the suspension of nanoparticles, here's a cryo-TEM. We can see that these are in the 100 to 200 nanometer region, and based on the DSC of the dry powder, it's amorphous. And these are what the spray-dried powders look like, as well as some dissolution and resuspension data that suggested that these were going to perform well in vivo. And 2 in vivo studies were done, one in dogs where we looked at the nanoparticle suspension, a nanoparticle OPC, which is basically the dry powder resuspended, and then the commercial capsule. And the dog pharmacokinetic data for the nanoparticle suspension suggested that we're getting much higher absorption, which you'd expect for the amorphous versus the commercial capsule. And in the clinical study, one thing to point out, again, here we're looking at a commercial capsule, the resuspended nanoparticles, and then we also did an SDD leg here to see, SDD versus nanoparticles, what's the performance. And what's interesting here is that the C efficacious for this drug is somewhere right around 300 to 400 nanogram per mil plasma concentration. And these look fairly similar in terms of rapid onset, but the nanoparticles were pretty close to 10 minutes for C efficacious versus an hour for the commercial capsule. And for a pain indication, that could be significant. So the third case study, this was a very challenging spray-dried dispersion. You can see it's over here in what we call the brick dust regime, and has peptidomimetic qualities, and this is an indication for rheumatoid arthritis. So effectively looked at this, screened several 10% loaded dispersions just because this is where it belonged in the map. And we had low solubility in acetone, but for this feasibility, that was deemed acceptable. Spray dried the product, showed that it was amorphous, and that we're getting these typical shriveled raisins. And in this is a dissolution test where we use centrifugation to kind of separate out the drug species. And if you look at the API, we're getting significant enhancement, and again, looking at only 10% active SDDs. And effectively, for this dissolution test, the area under the curve is containing things like drug polymer nanoparticle colloids, free individual dissolved drug molecules, and then drug that's partitioned into bile salt micelles. And at first blush here, you know, we would look at this and we'd say, okay, so PBPDA has the highest, you know, exposure or dissolution extent. That should be what we select. When in fact, if we go do a separate centrifugation step where we get rid of these species and we only focus on the most mobile active species, which we believe are most important for absorption, that's shown here. And while they're close, the HPMCS-M-based SDD gets the highest free drug, and so that was what was selected to go into in vivo study. And effectively, this was a PD. This was an animal model, arthritic animal model. So they give the animals arthritis and then dose at each of these black data points during the study. And what you see for the HPMCS SDD is that this plateaus. This levels off, whereas the control group continues to progress their disease. So case study 4 is a lipid-based case study, and this was a compound that I thought was interesting from the standpoint that, you know, at first blush, looking at these compound properties, you know, especially a 300 milligram dose, and, you know, not a very high log P, you know, you might question if this was the appropriate technology selection here. But in fact, as you'll see, because the technology works so well, the dose was able to come down, and then it suddenly did become a very, very good candidate for what they were trying to do with this program, especially given the food effect and some of the other metabolism and efflux issues that they had. And so the in vivo data here, 4 formulations, 3 self-emulsifying, 1 aqueous suspension, were dosed in fasted dogs at 30 mg at the conclusion of the 10-week program. So again, pretty quickly getting into animals. And then all of the lipid formulations were tested showed an increase. And probably the most important here was that at 30 mg, the AUC values were higher than that of the API at the capsule dose at 300. So here's a case where because the technology is enabling and providing improved bioavailability, you know, what was originally thought might be a stretch for the technology actually put it quite a bit into the sweet spot. So for a last kind of case study, this is a couple of slides on the solid lipid particle example, and this is in an OTC product, and effectively multiple controls were run against a reference, and you know, effectively what we have here is the blue is the immediate release SLP, and then by playing with the formulation slightly, we can change the release rate of the drug coming out of the SLP. And if we take these formulations and put them into beagle dogs, we see it correlate, the release rate correlate to the exposure, which is kind of a neat thing to be able to do in terms of tuning for immediate release or modified release, as well as just getting in the immediate release case, getting bioenhancement. A second example in the SLP area would be this health and nutrition example, looking at lutein and the zeaxanthin. And again, this is an example where massive bioenhancement for these two in these two cases. And the zeaxanthin's not shown, but it also had similar enhancement relative to the reference products. So I think with that, I'd like to just follow up on a few conclusions here and in this morning or afternoon, depending on where you are. So I think the conclusions to come away with this real high-level summary overview are there's a large number of bioavailability enhancing technology options, and it's at almost too many to the point where I think people can sometimes be, you know, not know what to pick, and so a lot of what we talked about today is how do we get over that? How do we make the right thing, the right problem statement up with the right technology? Everything we talked about today but besides the SLP example, has precedence from preclinical scale through commercial product, which I think is really important when you're selecting technology. It's always nicer to know that there's a commercial endpoint that it can go through, and that you're working with people that have that breadth and experience to know what it takes to go from formulation selection to commercial manufacturing. Yeah, as I mentioned, it's important to kind of select and tailor the technology to the API properties, but also other things like the dose and the product concept. Sometimes marketing gets to weigh in. It's a multi-dimensional space. The approach that we're taking is to use historical experience in the form of these guidance maps that we talked through, and use that to say, when we get a compound with properties and a dose, and try to weigh all the pieces of the programs that we had seen, what's the right technology based on this map, and use that to kind of narrow down the number of formulations we screen early on, so that we can be more efficient with the resource and the timing. And for us, the selection process is multidimensional, but we're making progress towards the holy grail of really intentionally selecting the technology based on the as I said, based on the product concept. So with that, I'd like to thank everyone for tuning in and listening, and be happy to do my best to answer any questions that people might have.
Great. Well, thank you, Dr. Vodak. That was very interesting. I would now like to invite our audience to continue sending their questions and comments in right now using that questions window for this, which is the Q&A portion of the webinar. And Dr. Vodak, I've already received some questions, so I'm going to start with those. Our first question here: what guides choice of SEDs versus SLPs, the solid lipid pellets?
This is going to sound a little bit like a cop-out of an answer, but I think right now we're in the process of evaluating what compounds and product problem statements are appropriate for the solid lipid particles. And so at this point, I don't think I could answer that beyond what we had talked about in the presentation of what would guide the selection of the SEDs.
Okay, thank you. Our next question here: what determines selection of loading in spray-dried dispersions?
Well, as I tried to cover with the spray-dried dispersion map, the two things that determine the selection of the loading in an SDD are the melting point to glass transition temperature ratio and the LogP. So things that have a high TM over TG ratio are typically much harder to stabilize in the amorphous state, and therefore you have to dilute it out with more of the high TG polymer. And on the other side of things that are very greasy and lipophilic can have dissolution rate issues, and so you have to limit your loading of that greasy compound in order to have something that continues to dissolve, as was exemplified in the Keras SDD case, where we knew right away that it was going to be hard to have much higher than a 10% active loaded system.
Right. Okay. Thank you. Our next question: any examples of bottom-up nanocrystals?
We do have some examples. I felt like I didn't have enough time in the scope of this overview summary, and I think our intention is to have a completely devoted presentation as part of this series of seminars around nanoparticles in general, and I think we'll cover it in that.
Great. Okay. Thank you. Our next question here: can the same formulations be used for spray-dried dispersion and hot melt extrusion? Sorry, I missed part of that question there. Can the same formulations be used for SDD and HME processing for amorphous formulations?
I guess it's possible. Typically, the types of polymers that you would want to use for an HME tend to be things that have better thermal stability than the cellulosics. Things like the PDP, PBPBA, those types of systems are typically more, but you can also make spray-dried dispersions out of those. The only difference I would say, based on the process, is that due to the different quenching rates, sometimes it's harder to get a metastable, stabilized amorphous system in an HME.
Okay, thank you. Another question here. Spray-dried dispersions and hot melt extrusions are both amorphous dispersions and fall in the same formulation space in your map. How does one select which technology to use in that space?
That gets to be in some of these non-represented, typically non-represented aspects. So if it's going to be a very high throughput process or has poor organic solubility, it might swing it one way or the other. Sometimes if the client's capitalized for HME but not for spray drying, that will drive it one way. The dose can sometimes move things around. So there's multiple things that can weigh in on what types of technologies we want to use.
Okay, thank you. And I believe this next question was referring to when you were talking about the spray-dried dispersions. This audience member says the polymers are highly water-soluble. Is that correct?
Highly is kind of a subjective term as well as soluble, I guess. I think the polymers that we're using, so you could have a whole webinar on just the polymers that you would use and select. And I think one of the nice things about some of the ionic polymers that we talked about is that in the solid state they're protonated and relatively hydrophobic, which limits the amount of water that formulations pick up and therefore can lead to better physical stability, whereas some of the neutral polymers may pick up more water. In all cases, when the materials hit pH 6.5, they should rapidly dissolve into, at the very least, colloidal systems where the drug's able to release or interact in a high energy fashion.
Right, okay, thank you. Our next question, does Bend and Capsugel have capability and commercial precedence in all the technologies you presented today?
I don't think I would say commercial precedence in that in everything that we presented we've manufactured commercial supplies of the technology. I would say more that at the very least we have progress toward the intention of having that, and in most cases have taken those technologies to very, very late stage clinical trial kind of commercial scale.
Okay, thank you. Our next question. Any comment on using supercritical fluid technology for particle engineering and bioavailability enhancement?
That's a good one. From historically 10, 15 years ago, we evaluated some of those supercritical fluid technologies and just felt that they weren't quite ready to be that kind of scalable, progressible technology. I think obviously 10, 15 years later there's been some great technology advancements, and we at Bend are even kind of starting to get towards some of that where we're superheating organic solvents in line to get extra solubility for different applications, including bringing up throughput. So that's one that I would probably haven't done my homework on as well, and probably should go read some more. But I think that there's some promise in that, and especially as the technology matures.
Okay, thank you. Our next question. For spray-dried dispersions, is the TG for the TM-TG relationship the glass transition temperature of the polymer or the small molecule?
Great question. It's for the molecule. So when we look at that map, we're not really thinking about polymers. We're thinking about the specific properties of the API. So when an API shows up, if you measure, you can calculate LogP, and you can run a DSC melt quench scan and generate all the parameters pretty simply and quickly, and then look at that map and kind of look at the different shaded regions, and then decide, okay, what else? Does this polymer, does this active have, is it acid hydrolysis lability in terms of we don't want it to be with an acid polymer? Okay, well then that'll help us select our polymer choice. So the map is specific to the API rather than anything with the polymer. The polymer can be kind of a or I guess probably a third dimension or fourth dimension out of that map.
Right, okay. Thank you. Next question. In case study 1, is CD necessary in all formulations? Can you use HPMCAS and get the same effect?
You can get the same effect of that initial spike and crash, but I believe that without the CD you won't get there'll be a small enhancement out at like 90 minutes that would be relative. Unfortunately, the patent application that I took it out of, they didn't have that part in an example. So I don't know if they tried that, but I would expect that it's probably not necessary for the spike and crash that you see in the first 30 minutes, that enhancement, but potentially for the solubilities observed later on.
Okay, thank you. Our next question here. This audience member missed the earlier part when you were talking about the SLP technology. For those audience members, can you just give a brief description of that for them?
Yeah, the SLP technology is essentially a lipid particle where the drug is either dissolved or suspended inside of the particle, and no drug is on the surface of the particle. And there's obviously lots of handles to tune in terms of the excipient that can go into the lipid inside of the particle and how it comes apart. And as I said, we're in the process of evaluating that technology, and we think it has a great deal of promise.
Right, okay, thank you. Our next question. These are a few marketed solid dispersions. How do we currently test the stability of these amorphous dispersions?
Good question, and I think that as we go to the next kind of deep dives in the technology, we'll really go into it. And so if interested, I recommend tuning into that. But just to try to answer your question briefly, what we typically do in terms of amorphous systems, especially in the case of, I think how you think about physical stability depends on what type of system you have. If you have a kinetically stabilized system, you can really think about what the relationship of the glass transition temperature is to your storage condition. If you're thinking more around a thermodynamically stabilized system, now that's a solubility, so that can be a little bit different. But if we just focus on the kinetically stabilized system, we can say, basically we know if your system has mobility, it will convert to a lower energy form. And so you can essentially look at how much mobility by where your storage temperature is relative to your glass transition temperature of your dispersion. And then based on that, you can generate the kinetics that will tell you how fast something's going to crystallize at a given storage condition. And so you can do everything from just basically say, I want my TG to always be 30 degrees higher than my storage temperature. That's a fine kind of rule of thumb. You can do the intermediate kind of predictive physical stability where you're actually generating crystallization kinetics and extrapolating out to longer time points. And then, of course, there's the empirical, let's put some stuff up on stability, although we feel pretty good that we're not going to be surprised because we did the other work.
Right. Okay, thank you. I think we have time for one more question here. Which is a better solvent for stabilizing solid dispersions like pH-altered water or an organic solvent? Is there any preference for a non-aqueous solvent?
Okay, so I'm going to interpret that question to mean stabilizing solid dispersions as a suspension for dosing. And try to answer it that way. I don't know if there's a way to ask the questioner if that's what they mean. Definitely with suspensions, we've had more success, and probably just because of where our clients have preference of using pH altered water buffers, and sometimes even adding in some things like methylcellulose. And where it gets to on the suspension preparation is you have to understand where your failure modes are. So if you have a really low TG dispersion and you put it into a suspension, potentially the water ingress can cause phase separation and crystallization inside the particle because you have mobility. On the other side of things, if you have reasonably high solubility in your aqueous media, you run the risk, or especially if you're putting things like surfactants in, you can leach drug out, and now you can run the risk of ripening. And so depending on what problem you're trying to solve in the suspension preparation would drive towards what you've used. And I'll just say in really desperate cases, we've done things like try to make suspensions in olive oil and things like that. But that's certainly the exception rather than the rule.
Right. Okay. Well, thanks for that. And thank you to the audience for all those great questions. We have reached the end of the question and answer portion of this webinar. If you do have any further questions, however, you can direct them to Alyssa Barrett at info@bendresearch.com, or you can phone 1-800-706-8655. And thank you, everyone, for participating in today's conference. You will be receiving a follow-up email from X-Talks with access to the recorded archive for this event. And a survey window will be popping up on your screen, and your participation is appreciated, as it will help us improve our further webinars. Now, please join us in thanking our speaker, Dr. David Vodak. We hope that you found this conference informative today. Have a great day, everyone.